I want to first thank you all for navigating through the maze that we set for you. That's one of our scientific studies that we do at the NRI. We tell you to show up on a specific corner at a specific time, and then we see how long it takes you to get around all the construction. So, so job well done, and thank you very much for making the extra effort to get here. This program tonight is the close of our 2018-2019 season of Appetite for Life. And it's the culmination of our 10th anniversary year as a research institute. So it's especially special to us, and we're really thrilled that you're here to share it with us. I want to make sure that I thank Restaurant 46, as always, for hosting us. They always do a fine job. I hope everybody's had a chance to grab some food and relax and enjoy. And before we get the program started with our speaker tonight, Dr. Trujillo, I'm going to give the mic to our director, Dr. Steve Zeisel, to introduce her. And I do want to say that after the program, please stay for just a moment for some more comments.
Sorry. It's painful. So again, thank you for coming tonight. And again, you're one of the reasons that we're encouraged about being here and working in a community that cares enough about what we do to show up over and over again to hear some of our progress. Tonight, we're very fortunate to have who came to us from Mexico and has been training here at the NRI for, it's 4 years now? 4 years today. And one of the purposes of the Nutrition Research Institute is to create an opportunity for scientists from all over the world to come to North Carolina and learn some of the approaches that we take and then take it back home. And in a year or so, Dr. Trujillo will be going back to a faculty position, I'm sure, in Mexico and create a research team that we in North Carolina can continue to work with. And tonight, she is going to talk to us a little bit about the project that she's working on now. And hopefully you'll ask her some questions and learn about how it could involve you as well, but also learn about how much she feels she's learned and why this is an internationally recognized research program that we can all be proud of. So thank you, and then after the talk, you'll hear some comments, but most importantly, some cake and champagne to celebrate our 10th anniversary. So I'll give you Dr. Trujillo, and I'm sure you'll have a great time. You don't need this?
Okay, well, thank you all for coming today. It's my pleasure to present what we have been doing. Today I'm going to talk about a lot of fun stuff that we are doing in the lab, science that is happening right now at the NRI, and I hope that you enjoy this talk. So first of all, I'm going to introduce myself a little bit. You notice that I have an accent, right? So I'm actually from the south of Mexico. It's a place near to the beach, and it's called Hotland. So usually the weather there is around the 90s. So it's really hot. Then I moved to Mexico City, and I studied biology. And I like that because biology is broad. So I study animals, I study cells, I studied plants. So it was a lot of fun. But since then I started to develop an interest about research. And back then I was interested in how our medicine or the pills that we take different depending on our genetics. So that was my main interest. But then I did my PhD in another institute, and I was working with a vitamin that also had some interesting interactions in our body. Then I wanted more, and I wanted to do more things and to do other type of research, and I found Dr. Seisel Lab, who's been working for 30 years, I guess, about choline. So my focus in his lab is to understand choline and personalized nutrition. So I like this picture a lot because actually I love this guy here. It's clean, not like back home, and the sunsets are great. So first of all, I said choline like 10 times, and probably some of you don't know what choline is. Well, choline is a nutrient that we can find in meat, we can find it in fish, some vegetables, in chicken, and in milk, cheese, and eggs. I know that we scientists have been confusing you. Years ago, we said, no, do not eat eggs, are bad for you. They have cholesterol. And then we came back later and said, wait, there's good cholesterol, there's bad cholesterol, so you can eat only the whites. So now, also, research that is going on at the NRI with Dr. Katie Mayer found that actually it's not good to eat eggs if you have some heart problems or some cardiovascular disease. If you are healthy, you can eat eggs and you will be having enough choline. So, but not only we can get choline from the food, we can also get it from our body. So our body make choline for ourselves. And this happens in our liver. And this is through a gene that it's called, it's this long name, but I'm just gonna say PEMT, will be easier for all of us. So, and we can get phosphatidylcholine. So phosphatidylcholine, it's very important for our cells. All our cells have a barrier that can transport stuff in and out of the cell. That is called a membrane, and that membrane is formed from phosphatidylcholine. So we get choline from the food, and we can also make choline in our bodies, depending on our genetics, right? But what is choline really doing? So tonight I'm going to show you some evidence about the importance of choline in our bodies. So in liver, I'm going to show you some images about how low choline can cause fatty liver. I'm going to show you some evidence about choline and muscle. I'm going to show you that it's essential for brain development and that it's important for memory. So, as I said, if it's, sorry about that. As I said, it's really important to have choline from the food. And we are also making choline ourselves. So probably now you are wondering, am I eating enough choline if it's important for all those things? And the other point, and really important, can we all make choline in the same amounts? And the answer, I will show you some of those answers. So first, are we eating enough choline? Short answer is no. You can see here in the purple bars, it's the adequate intake, what is recommended. And here in blue, it's the actual intake. So you can see that we are eating less choline than we should, okay? And the other thing, remember that I mentioned that we can make choline from ourselves? Happens that not all of us can we do that. And this is why. We have in our DNA some DNA differences, some misspellings. It's like when you put your password to enter to your email, if you misspell something, you cannot get access to your email, right? And then you need to retype exactly the words. So something similar happened with our DNA. So there are codes in our DNA. And if there are misspellings, we are not getting the result. And we have we can have these variations. Instead of having an A, some people have a G, some people have a T. So, but the thing is that we all have these misspellings, don't get me wrong. The thing is where those misspellings are. Sometimes are in important genes. And in fact, in the NRI, we have a PI, Dr. Martin Kohlmeier, who's here in the back of the room, who is working on this, about how genetic variants in our DNA make us different and make us get nutrients in a different way. So basically, a SNP in PEMT can make us not make enough choline ourselves, or de novo, that it's called. So let's say you are not eating enough choline, and chances are that you may have a SNP or a variant in PEMT, then you are really low on choline, because you cannot make enough, and then you are not eating enough. So knowing that you have a SNP can make you actually eat more choline, and then you are compensating, and you will have a healthy life. So, but how do we know that choline is really important? We know because we have models. And what a model is, it's something where we can mimic the actual phenomena, something where we can reproduce what is happening, and then we can test it. So I'm going to show you some work with some models that we have been doing. And I'm going to start with pregnant woman, because that's really important. It's not only what the mom is having, it's also having the baby and the effects on the baby. So to study that, we are using mice. So we have pregnant mice, and they are in adequate choline diet, are in low choline diet. We change that diet during neurogenesis. Neurogenesis is when all your neurons will be formed. Okay? So it's a really important period of time. And this is happening in mice between embryonic day 11 and embryonic day 17. In humans, it's more or less the first trimester, right? 25 weeks. So it's the process where so what you're seeing here is the cerebral cortex of an embryonic day 17 pop. So the cerebral cortex is the very top part of our brain. It's where part of the memory, it's part of visual memory too. So it's really important. It can also help us to process language. So what you're seeing here in green, these are the cells that are called progenitor cells. These cells are the ones that will give rise to all the neurons and other cell types to form the cerebral cortex. Yeah, okay.
Battery's dying.
Okay, so I'm going to go over this again very quickly. So this is the cerebral cortex of our embryonic day 17 pop. So this pop was in adequate choline diet from embryonic day 11 to embryonic day 17. And what you are seeing here in green are the cells that will give rise to all the neurons. What happened in low choline is that you have a reduction in these green cells, which means that at the end, these mice won't have enough. And then we wonder, okay, so these cells are not enough. What is happening? So we take a look inside here, closer, and this is a zoom of the cerebral cortex. And what you can see here in green, in red, are cells that are dividing. Are cells that are dividing because then they will decide to go to neurons. So you need to have enough to have all these circuits. And what happens in low choline is that you don't have that many cells proliferating. So this is like very clear, I guess, for you too. You can see the effect there. And then we thought, well, if these cells are having some issues dividing, and these cells, these neural progenitor cells, are also in the developing eye, what is happening in the developing eye? So here's a picture of an adequate choline pop, again E17, and you can see here this structure. Here is the retina. So basically it's in the back of the eye, and it's where the light hits. Think about it as a camera, and then you will process the images and you will see different colors. This structure has different cell types, and you can see that it's really, really tight. What happened in low choline is that some pops have these convolutions. The structure is not correct. And also, there are some spilling of the cells out of the area of the retina. So actually, this is a paper that we recently have accepted, and where we explain a little bit more about what is going on. We took a look closer to that, and I hope that you can see here the green cells. These are neurons in our eyes, and here in magenta are the cells that are dividing. And if you look here, this is how the eye looks like, and the neurons are not actually in the layer that they should be. So this is really important and really interesting, and we are seeing a phenotype. We are seeing something that is happening. But actually, what are the other things that where we can see the effect of these cellular changes? Because someone can tell me, yeah, but if the pop can still see, what is the problem? Or if the other eye is compensating, what is the problem? Well, happened that some other studies in mice, this is a behavioral study. So what we did is to check months later how these pops, no, this is not us, it's other group, how they were behaving. And they have this test where they have food at the end of the arm. So they go there and they grab the food. They have to remember that there's no food there because they already grab it. Okay? So basically, the mice that had extra choline made less errors than the ones with no supplementation. So this is having an effect in behavioral stuff. This is actually some changes that we can see not only at cellular level, also in terms of behavior. And actually now at the NRI, there's also another PI who is doing a bunch of behavioral tests for fetal alcohol spectrum disorder and choline supplementation, and she's always here. She's also here, Sandra Mooney. So if you want to talk more about it, you can talk with her. So now you can say, okay, this is all in mice. Yeah, right? So how do I care about it? So I'm going to show you some evidence very quickly about what is going on in humans. So it happens that adequate choline during pregnancy reduces neural tube defects. So you know that all the pregnant women are having folate supplementation, right? It's like something that it's going on. Like you need to eat folate. You need to take your folate pills. And that's vitamins, sorry. And that is because the development of the neural system depends on folate and methylation. That's something that you don't need to remember, the methylation part. So, and, but some people, even if they are eating enough choline, enough folate, they are not eating enough choline. So their babies can have some defects. So folate, in some cases, it's not enough to have a good, a healthy baby. So also someone at the NRI who is working with folate and some genetic stuff, it's Dr. Sergey and Dr. Natalia Krupenko. So they are doing research in the side of folate. So another study, this is a big study from Harvard. They check the intake of pregnant moms, and then they check their babies through time. And they saw that at 7 years old, the babies have better visual memory, the ones that their moms were eating enough choline. How do they know? The moms go, answer a questionnaire, and they basically put in figures what they are eating. So that's how you can more or less calculate the amount of choline that people are eating. So this is in development, this is for trials, but what's going on in adults and what's going on in men? So also, higher choline was associated with better cognitive performance in adults. So that means that they can remember things, that they can give details of a picture. In fact, you remember more than you think that you remember. If you have a picture of today and I show you and I move some stuff, I'm sure that you will remember exactly what I moved, even if you think that you are not really storing that information. So also choline is important in disease. And for that, I want to talk about this study where choline intake prevents the decline associated with Alzheimer's. And this is in humans, so it helps with memory. You will have sharper memory. And it was recently another study, it's also in mouse, but it's important too, where moms and dads were supplemented with choline, and that protects not only their babies, it also protects the next generation. So this is also something interesting. So these all have been, I've been talking about brain development, cognition, but what is going on in other organs, right? So I'm going to show you some images again about a model that we have. So this is a genetic model. What does it mean? Is that we remove some of the genes and we see what happens. That's the way to test, right? So this is a mouse where we remove one of the most important enzymes in choline metabolism. And what we observe is that these mice develop fatty liver. So and you can see here the fat. I was hesitating about putting these pictures, but I guess it illustrates enough. So, and actually, remember that gene that I mentioned at the beginning, PEMT, the one that helped us to make choline from scratch? Well, we observed recently, and that's not published yet, that's only an observation that we made in the lab, that also the mouse model lacking of this gene also develops fatty liver. So that's something that we are really interested in follow-up. But I want to show you that this is not only happening in mouse. So in patients that were hospitalized and they were having parenteral nutrition, that means that they have all the nutrition intravenously because they cannot eat or they cannot absorb the nutrients enough, so they need to put the food through the vein, intravenously, right? Yeah. So they started to notice that some patients start to develop fatty liver. That fatty liver was reversed when they were given choline back. So that's also evidence that choline is important. And also, this is in liver, but not less important, and in terms of the genetics, it's muscle. This is a study that was done here at the NRI, where males were put to do some sort of exercise. They also report what they have been eating, so that's how we can calculate the choline intake. And these studies show that the people who was reporting having more pain or some enzymes elevated due to the exercise was because they were not eating enough choline. And I guess that study is now extended a little bit to see how your genetics also is affecting this. So I guess I have convinced you enough that choline is important. And probably some of you are wondering now, how do I know if I'm eating enough choline? How much is enough? Well, that's the science that is developing now at the NRI, because so far there's no way to test for low choline. It's only when you have fatty liver or the disease when you can tell that you were eating less choline. But there's no way to test in between. So thinking about that, Dr. Seisel put together a team, a very interesting team with different points of view. So we all are working, trying to see how can we tell if you are not eating enough choline. But we don't want to catch you when you are really eating low choline. So we want to have like different points, right? So I'm gonna talk a little bit about this study. So we are recruiting males and females from 17 to 70 years old. The participation is up to 6 weeks, and participants will be compensated. There are some restrictions, of course: no smoking, no substance abuse, no overweight, no unusual diets, and no history of liver or kidney disease. And if you have liver or kidney disease and you are eating low choline, we don't want that. Okay? So the design of the study is this. So basically, for 2 weeks, we are going to provide the diet to the participants. We are actually doing this. We provide a diet. And after that, we give you a Choline D9. Choline D9, it's safe, it's FDA approved, it's same as choline, but it's tagged. It's a way for us to follow up what it is. It's a way to us to see it. And we do some experiments with that. And participants give us samples, plasma sample, urine sample, and we test some other things just to know that they are okay to enter in the study, and also we check them through the whole study. After your first 2 weeks, participants go 2 weeks, have a washout, can eat whatever they want, and they come back and they have a 50% choline diet or a 25% choline diet. Of course, they don't know in which diet they are, right? And we don't know either, so it's a blinded study. Only 2 persons in our study, they know how much choline they are eating. And we are checking labs all the time to make sure that our participants are fine. So the food that we are providing, I'm going to show you, because it's food that we eat like constantly, right? So these are some examples for breakfast. So they can have croissants, they can have some muffins, and for dinner they can have lasagna, pizza, and my favorite one, enchilada verde, of course, right? So here are the things that we check, okay, when you are entering the study and through the study. We need to check your heart rate just to be sure that you are okay. And this is really easy. There's a band. The participants put their fingers, and a little iPad gives you the reading. So it's really easy to do. Also, participants provide us of a saliva sample. And this is to check participants' DNA. Because remember, we want to know if you are in high or low choline, what are the outcomes, but also how your genetics plays a role in this. So participants will provide us some plasma sample also to check some liver levels and some other things that we need to be checking through the whole study. We do pregnancy test. Of course, we don't want to have pregnant women eating low choline if we know that the outcome might not be good. And you sit there and just fill the questionnaire about what you've been eating before entering to the study. We also do an ultrasound just to check that you don't have any abnormalities. And we do fiber scan. And this is actually me, and this is Rick, who volunteered himself to take this picture. And with this, we check, With this, we check that you don't have fatty liver or how much fat you have in your liver. It's not invasive. It's not like a biopsy that it's painful and bloody, and nobody wants to do that. So this is actually just, you just feel like a little vibration. So it's really easy to do. And we also are doing the muscle part. We want to know if we put them to do some exercise, what are the outcomes, right? And then you travel to Wake Forest to have an MRI. So that will be in your liver, an MRI in your liver. So with all of these, having your genetics, having FibroScan, your labs, your muscle. We want to see what is the best exam to see what is your choline level. So with all of this, probably in the near future, you can go to the doctor and say, you know what, add to my panel the choline because I want to know if I'm eating enough choline. So if you want to join our study or know someone who wants to join the study, this is the phone. It's wrong in the flyer. So if you want to write down, this is the number, and you can contact Julie. Julie is right there. She's the study coordinator. She keeps all of us on track, and also she organizing our running club. So you can see her here. I just want to thank you all for coming today. I want to thank my boss who gave me this opportunity to be part of this amazing group. This is an old picture. We were to Asheville, all of us. We talk about science all the time. And this is the people that are involved now in the study. And I really want to thank the people in my lab because really they are who challenge each other and we can come with good ideas. And thank you for coming tonight. So, if you have some questions, I'm happy to answer. Yes.
As far as the eggs are concerned, is the choline part of the egg or in the yolk?
The yolk.
That's what I thought.
Yes. Yeah, but if you don't have any cardiovascular disease, you can eat 2 eggs and you will be eating enough choline.
Okay. Per day?
2 eggs, yes, per day. But if you want to check other foods, actually you can Google only like choline and FDA, and our lab together with the USDA. Yes, I was about to say FBA. I don't know. No, USDA. So we made a list of different foods and the amount of choline. So if you want to check, you can always have that PDF and you can check the amount of choline in vegetables, in some other food. Mm-hmm. Yes. Well, I couldn't dare to say to someone now to do supplementation without knowing your intake. Now let's say that you want to do supplementation. I will say that it's safe, and the only adverse effect that people have reported, it's like having fishy odor. So that's the only thing. Mm-hmm. Yes. Actually, you can reuse choline. It's not reported what will happen if you eat a lot. Maybe you want to jump on that?
Sure, I'll jump on that. So the amount you need is half a gram a day. And it's okay up to a few grams a day, but much more than that has side effects. So you don't want to take 10 times as much, but about half a gram to a gram a day is plenty. So those pills are about a half a gram a day. 2 pills is more than you need, and 1 egg is about a quarter of a gram a day. So that's a quarter of a gram. So you need 2 of those pills of that brand pill to get the half a gram you need.
Everything else we go through, what we're really getting is that's not FDA approved. Taking the chances.
So most of the choline in the health food stores contains the choline it says it has. But it's very easy to eat foods. Eat cheesecake is a lot more pleasant
Okay. Yeah, sure.
You may have touched on this a little bit.
Sorry, can you repeat that?
Is it in the scope of your project to determine why choline does this or how it does this?
Yeah, of course, it's part of our project. Um, um, I didn't want to enter into all those molecular details, but of course we know how it happened. And we use models too. And for instance, for the brain part that we did, we know exactly what is going on. And it's mediated by a non-coding RNA. And that's part of our genome. Years ago, we thought that that was doing nothing, but it happened that it's codifying and that it has a role. So it's a little piece that it's actually making the cells not divide enough. And for the retina, it's another different mechanism. So of course, we do that. We want to know exactly what is happening at molecular level.
What other foods besides egg? You had mentioned meats,
Mhm.
and what is
Fish.
the amount in fish?
Mhm.
What do you get from, how much choline do you get from that?
Yeah.
So I think as Dr. Trujillo told you, if you just Google USDA choline, on the U.S. Department of Agriculture's website, they have a table of 1,800 foods and how much they contain. So you can look at every part of your diet and figure out what are the good sources. So if you're a vegetarian, for instance, there are certain, wheat bran, wheat germ, where you can get a reasonable amount of choline from that. But since there are 1,800 foods, it's hard to go through each of them now. You can look it up, USDA choline, and it'll kick out that table for you.
Thank you.
Do you know if there are foods that make it more bioavailable for our bodies or that block our production of it?
No, because actually when you eat the food, it will be break in your by your microbiome. So you know all the bugs that we have. So actually that is what will make it viable. So the breaking. So that's other part of the study that we are not sure that we are going now. So we have been talking about it like more specifically about how your microbiome have also an effect, because actually it will break down.
So there's not a food that will block your production?
No, I will say no. Mhm.
Now, I think one thing that Dr. Trujillo didn't fully mention to you is that only women with estrogen make a lot of choline themselves. And men and postmenopausal women are much more dependent on their diets because estrogen turns on the gene in the liver that lets a woman do this so that she can have a good baby. And so if you've not got estrogen anymore, or never had it if you're a man, then you might have to be more careful about what you eat.
Right.
Are there any more questions?
So
Oh, here's one back here.
Is there a certain number of people that you're expecting to end up in this study?
Yes, we promised to the NIH that at least we will have 100. And of course, the reason is because the more representation that we have, the better results, right? If we have 3 people, that cannot actually reflect what is really going on. So far, how many do we have so far, Julie?
Depends on where we are in the study. We're constantly enrolling, so we'll be doing this over the next couple of years. We have had over 30 enrolled thus far, and we have people in different phases of the study right now.
and what's the grand sum of money that they get paid for participating?
$2,400 for completing the study.
So that's a nice vacation.
We'll see if we're repeated.
Okay. It's up to 6 weeks, so yeah, with weeks in between for the washout. Yes.
What's wrong with men over 70?
There's nothing wrong. So, but we just don't want to put people under the stress or not having a proper diet. That's the only thing.
I think to help you, we had to choose an age group. And so we chose an age group
much.